Thank you, everybody, for coming back into the room on time. So I think we really should start with the second talk of this session. This is another very exciting talk given by Dr. Elissa Epel. She's from the University of California, San Francisco, and really her work is mainly on behavioral, social cycle factors that will affect telomere length and other aspects of longevity and lifestyles. So her talk, I think, the title is over there, is Lifespan and Psychosocial Factors Associated with Telomere Length. And Alyssa, it's all yours, and I hope that you can stay on time so that we can ask you a couple of questions. And also, she promised us that she will come back for the discussion session, which is very gracious of her. Thank you.
Thank you. Thank you so much for including me. I don't see my slides being projected. So the first slide should have my disclosure on it, but I don't see that. So
we can see your slides, Alyssa.
Does it have the disclosure on it?
Yes.
Okay, good. So
your slides are fine, your disclosures are on, so if you want to advance the slide, you just say next slide, maybe that will help.
Well, actually, I'm controlling the slides here. Okay, so I'm projecting my slides. That's what should be showing, not the old slide deck that you have. But on the webcast, I can see that you have. It's not the slides that I'm projecting on my own computer. So is someone trying to fix that?
Yeah, someone's trying to fix it.
Alyssa, we're trying to fix that. So also you can look at what's on your screen on the WebEx or the web conferencing software. We just don't have the right slide deck on the webcast, which is different. So that'll be up in a second. So if you want to look at the WebEx page instead of the webcast, if you're capable, if that's possible. Otherwise, the webcast slides will be updated in just a minute. We're going to be videoing the screen. Just 1 minute, Alyssa.
Okay.
Okay, so the webcast should now be showing the correct slide.
Okay.
So if you should see the correct slide up if you want to go ahead, hopefully.
Okay, thanks so much.
Sorry about the technical difficulties.
Oh, no, that's okay. So everyone can hear me?
Yes, you're very clear.
Okay. Thank you for inviting me, and I wish I could be there in person. This is a fascinating topic. I'm starting off with a disclosure that I'm a co-founder of Telome Health. It's a measurement telomere measurement company, and none of the research that I'm presenting has any relationship with the company. Okay, so let me start by having us think about aging and how we measure biological aging. When we do this by, of course, physical appearance, which is just a phenotype and largely influenced by UV exposure. And the other way we measure this is we wait until we're midlife, and then we develop the clinical risk factors that are so common, and by 60 years old, more than the majority of people have at least one of these risk factors. So it's really important and helpful to track biomarkers of aging, and in particular those that are sensitive to environmental exposures relevant to this meeting. So telomere length looks like it is helpful. As Maria described, it's predictive of diseases of aging. And what I'm going to be showing you is how sensitive it is to environmental exposures. So what do we know about how telomeres are regulated beyond the genetics? Telomeres and telomerase can tend to decrease in response, of course, to cell replication, and so exposures to antigens and also other immune challenges like reactivation of latent viruses like Epstein-Barr virus or cytomegalomy. They're also very sensitive to the biochemical environment in the blood and in the cell. And in particular, we know a lot about oxidative stress and stress hormones, and that these both in vivo are associated with shorter telomeres, and in vitro actually dampen down telomerase or shorten telomeres. We don't know much at all about the exogenous chemical exposures. Dale Sandler will be reviewing that tiny literature later, but it's very likely that telomeres might show some promise of being an index of these exposures. So some of you might be thinking, why have a whole 20 minutes focused on stress? And I just wanted to mention that stress is an essential part of the exposome. And I probably don't need to define the exposome because I think this group and this committee are some of the key leaders who have exposed the exposome and made this concept more accessible to people. So the idea is that we need to measure these environmental exposures well, because that's the only way we're going to really understand how our genetic inheritance is expressed, which is through interaction with different environmental exposures. And these are the big determinants of gene expression, given that most genes have such low penetrance. So it's this GE interaction that we know we need to measure in a better way and focus on. And part of the obvious exposures are pollution and toxins, and then factors that from the outside that become endogenous, like poor nutrition and substance use and abuse. And then there's another set of exposures that we categorize in this very crude category of stress or stressors, and that is social stressors and psychological stress. So let me just briefly explain how those get under the skin. Different factors in our environment, like the level of organization in the home, the safety level of the neighborhood, the type of social relationships, and then exposures, things that happen, stressful events and trauma. All of these factors shape our perception of trust, safety, and vigilance, and how calibrated our stress response system needs to be, how elevated it needs to be. And these, when there are chronic adverse exposures, stressful environmental exposures, this increases psychological stress perception in the brain and then creates a biochemical environment of stress that, if chronic, can be as damaging, as large of an effect size as smoking. That's what some of the emerging data is showing. So really it can't be ignored, and it's going to be interacting with the epigenome and gene expression. And I'll, of course, just be focusing on the telomere part of that. story. So another new piece to understanding how telomeres are maintained in people through the lifespan is that they're shaped very dramatically very early in life, and that if we ignore these early factors, we won't really know what we're seeing when we measure telomere length later on. So I'm going to be talking not just about stress, but some of the early determinants of telomere length: fetal development, early childhood, trauma exposures, and exposures to adversity like low income, low warmth parenting, and parental divorce and substance use. So those are kind of the categories of adversity and trauma that have been measured. And lastly, I'll summarize for you what we know about adulthood stress. And health behavior is also very important, and I have the luxury of not having to cover that because Dale's going to cover that later. And I'm going to end just highlighting some of the important both opportunities to implement stress and telomere length measures in population-based studies where we have genetics, and also some of the pitfalls that have been that have made this field difficult to integrate across different results across different labs and studies. So what do we know about telomere length through the lifespan? We know that telomere length is largely, that it's very long when we're born, and then it decreases at a rapid rate in the first years of life. So here you see a graph where telomere length shows a very steep decline in the first 5 years of life, and then it really levels out throughout adulthood. And so it's these early years that are very influential in determining the trajectory of telomere maintenance throughout the lifespan. So what's happening in these early years that is so influential besides the normal pruning of the immune system? Now, most of the studies I'm going to be talking about are really looking at the immune system, since that's where we can most easily examine what's happening with telomere length. And there may be parallels to what's happening in other mitotic cells, since studies have shown some concordance across tissues and cell types. So this is a summary of some of the big factors that we know that affect telomere length. And I'll go into more detail on each of these studies, but here's really the main message and overview of this story about lifespan influences. The intrauterine environment, both maternal stress and nutrition, are probably having an immediate impact on telomere length at birth. In early life, we also know that telomere length in children is associated with socioeconomic status and metabolic health, like obesity level. We've mostly looked at telomere, as well as early trauma in early life. And most of the studies on disease have looked at telomere length in adulthood and have found that it's often correlated with disease and or predictive of diseases of aging. But it's this early life period that may be having a bigger influence on what we see in adulthood. And what you see here are two different trajectories, one where there's vulnerability in terms of the social and behavioral factors, and so there's no buffering from the inevitable life stressor exposures. And so you see at the end a woman who looks more frail, and then you see the other trajectory, which is someone equally exposed to stressors, but has some buffering and resilience from the environmental exposures, social connections, behavioral factors, healthy lifestyle, and psychological stress resilience. So one of the important pictures emerging in population health is that health is shaped in part, adult health is shaped in part in utero. And some of the factors that shape adult health are the mother's nutrition, excess weight gain, and stress level. These have shown to affect birth weight as well as birth outcomes and later health outcomes. So, for example, poor nutrition and low protein can lead to low birth weight, excess weight gain later in childhood, which is called catch-up growth, and then early onset of cardiovascular and metabolic diseases. So do these early influences affect the programming of telomere length? That's a new question. I'm going to show you a few studies that have started to look at that, and these are in collaboration with our colleagues at UC Irvine, Sonia Entringer and Pratik Wadhwa. So what do we know about transmission of telomere length? Well, there's large genetic influences at birth, around 50%. Now, what about the intrauterine environment? There have been a shockingly few studies using experimental models to understand regulation of telomere length in terms of intergenerational transmission, behavior, and social environments. Most of this work has been genetic. And so I want to point out a study by Suzanne Ozan and colleagues that actually manipulated the intrauterine environment and looked at telomere length and longevity in rat pups. So some of the rat pups had protein restriction while they were in utero, so prenatal growth restriction. They showed, they came out with low birth weight. They showed catch-up growth, like we see in humans, shorter telomere length, and early mortality. So the intrauterine poor nutrition actually looked like it had the typical effect of programming earlier obesity and mortality. The other group of rat pups had postnatal growth restrictions, so they had a normal fetal development, and then they were exposed to low protein. And these rats had actually longer telomere length and longer lifespans. So we know that at least in these rat pups, early nutrition is very important. Sonia Entringer has linked for the first time maternal folate levels to telomere length in newborns. So there's a clue that there's a whole area, there are a lot of relationships between maternal experience and nutrition and telomere length. We've also looked at telomere length in adults who reported that their mothers were exposed to severe stress. So in this study, Sonia had adults interview their mothers about their major life stressors like death and separation while they were pregnant. And all of these two matched samples, those healthy adults without prenatal stress and those with prenatal stress, were very, very healthy and well matched. And what you see here, these two plots, is that the prenatal stress exposure group has significantly shorter adult telomere length compared to the comparison group. And Sonia has recently looked at this in newborns. So rather than retrospectively looking at telomere length in adults and self-reports of maternal stress, she actually measured maternal anxiety during pregnancy and measured telomere length in cord blood and found that there was a striking relationship that was robust to covariates, showing that mothers who experienced more anxiety during pregnancy had infants with shorter telomere lengths. And that was just in press yesterday, so I'm so happy to be able to share that, because I think that it's important to look at what's happening right at birth. Now, what do we know about adults? There's been a handful of studies, there are now 6 studies, interviewing adults about childhood trauma or adversity. So now we're not talking about prenatal adversity, but early life adversity. And these 6 studies have found that adults who report greater childhood adversity have shorter telomere length as adults. So here you can see the first study on this. There were 10 adults who reported childhood adversity. Mostly it was emotional neglect. And then there was a group well-matched group who had no adversity. And you can see that even though there were no current diseases or depression, the group who had early adversity had significantly shorter telomere length. We've been looking at different clinical disorders. In one study, we looked at post-traumatic stress disorder. And we found that, not surprisingly, those with PTSD had significantly shorter telomere length than controls. But what was so interesting about this study is that we also measured childhood adversity, and this is where the real story emerged. You can see that those who had a critical threshold of childhood exposures, so those at the 4 and 5 end, multiple exposures, had significantly shorter telomere length. And when we compared the effect of PTSD versus trauma exposure, the effect of PTSD went away. When they were in the same model and we covaried trauma exposure, it was really trauma exposure that was predicting shorter telomere length. So this leads us to, ask how much of early experience is shaping what we see when we measure telomere length in adults. And one thing we've got to consider is these exposures don't just shorten telomere length at the time, and they might be shortening, some of the stem cell reserve, but what we know is happening is that exposures are shaping how our brain responds to stress throughout the lifespan, or at least they're shaping brain connectivity and volume of certain areas. So early experience shapes both certain parts of brain development and behavior. Nurturance, parental warmth, and good attachment appear to be protective to these developing areas of stress reactivity and motion regulation, and severe stress exposure appears to wire the brain to be more reactive. So what you can see here are some animal studies showing that chronic stress exposure leads to, if you look at the right-hand panel, it leads to expanded volume of the amygdala. And here you can see the dendritic branching, the pink neuron. It's has flourished under chronic stress, and this is the home of the limbic center and the alarm center in the brain. On the left panel, you see neurons from the prefrontal cortex area in the hippocampus, and chronic stress has the opposite effect on these neurons, so it's actually leading to atrophy and less branching. So what effect does that have throughout the lifetime? It may be affecting and creating exaggerated threat perception and stress reactivity. So it may be that part of the biological embedding that we see later in life that we think is related to stress might be also through changing our central control system, in a sense. So now I'm going to shift to what we know about stress and cell aging in adults. And I can pretty much summarize this pretty quickly. Thinking about measurement of stress, we can measure these objective situations like caregiving, events like exposure to violence. We measure perceived stress and threat appraisals, and then physiological biomarkers of stress. And chronic stress is associated with greater risk for certain diseases of aging like cardiovascular disease. Telomere length may be a mediator in this relationship since it's both predictive of diseases of aging and shortened by chronic stress. So here's just a laundry list of the various types of stressors that have been associated with shorter telomere length. So socioeconomic status, and in particular education level, later in life, decades after peak education was attained, is associated with shorter telomere length. This is some studies like Whitehall and Health ABC. Perceptions of stress in both younger and older samples is associated with shorter telomere length. And interestingly, threat appraisals alone, when you just assess in a standardized way how threatening people view a standardized lab stimuli, this is also associated with shorter telomeres. Being a caregiver, having major depression, exposure to adult traumas like domestic abuse, and then, as I mentioned, early life trauma. And there's an interesting new study by Shalev, Caspian, Moffitt showing that exposure in 5-year-olds, and they looked at cumulative exposure to violence up to 10 years old, so over a 5-year period, was associated with telomere shortening in buccal cells. So this shortening does appear to be happening early in life. We really don't know much about what's happening in other tissues, and in particular in the brain. There is a very interesting new animal study that did look at a chronic stress model. They used the model of uncontrollable stress, which we know can create depression, and they looked at telomerase in the hippocampus. they found that stress was damping down on telomerase in the hippocampus and blunting neurogenesis. They then also just looked at what happens when we block telomerase, and they get the same syndrome of impaired neurogenesis and depression. So telomerase may be a mediator in this relationship between stress and depression directly, in part through the brain levels. This is a study looking at duration of depression and telomere length by my colleague Owen Wolkowitz at UCSF. And here you can see that people who've had depression for a year or two, their telomere length is very scattered. There's no relationship between depression exposure and telomere length. It's only when we get to severe exposure, 7 years or more, those people are on the short end. So we see there's an exposure effect. I wanted to now just take a step back and think about, how can we use this information about going into the blood, looking at telomere length at one point, and learning about exposures. So most of this conference is focused on the green bubbles on top and the genetic and epigenetic control of telomere length. When we think about telomere length and how in vivo and how dynamic it is over the lifespan, we also need to think about the green bubbles there, the more behavioral, psychological, and biochemical. And I do want to note that this is a complex field, and some studies find longer telomeres with stress exposures, or in cancer case control studies, not shorter telomeres. And we really haven't cracked that mystery, but there are some clues that telomere lengthening happens in states of severe stress. So, for example, there's a very interesting study looking at exposure to severe oxidative stress in divers and finding that there is acute telomere lengthening over several months and then a dramatic telomere shortening. And what's probably happening there is a mobilization of new progenitor cells during the acute stress phase. So you see acute lengthening, and then you see a dramatic shortening. And this might, over time, be related to stem cell exhaustion and early senescence. If you have a repeated mobilization, you're using up these progenitor cell stores. So there are some, big questions there that can probably best be answered by animal studies. There are many opportunities to be adding some very easy and low-burden measures in some of these larger cohort studies where the other aspects of the exposome are measured. So I wanted to mention some of these. And in terms of early life, one can measure maternal stressors, and particularly during pregnancy, maternal chemical exposures, birth weight, and gestational age, both of which have been linked to shorter telomere length. Early adversity can be measured with, for example, 7-item questionnaires to get the really big events. Major life events and chronic situations like caregiving and work stress are easy to add. And, of course, stress perception, which matters and needs a few more items. We measure it with 10-item questionnaire. So stress measurement, it's not that easy, but it's probably worth doing because it's going to be, potential moderator of other exposures or of genetic endowment. And I just wanted to mention that NIA is working very hard on trying to reduce stress measurement to simple items that can be used in large cohort studies. So that's an ongoing effort led by Liz Nielsen. So that when we think about measurement of social stress, we have kind of the easy tier 1 measures, the exposures, which can be assessed with self-report and could be retrospective. Then we can assess perceptions of current stress, either in a cross-sectional way or with daily monitoring. And then there are the biomarkers, if we can get into blood, et cetera, and look at markers of stress, basal stress and reactivity, and, of course, telomere length. So it's relatively easy to measure telomere length. Most of the studies have looked at blood cells through blood draws, but we have also been piloting new studies that look like they're, making sense using dried blood spots and salivary telomere length, since saliva is mostly blood cells. Buccal cells could be used, cord blood in newborns. The collection issues are important. So if someone is ill, it will most likely change the distribution of cells present and telomere length. We try to measure it in standardized conditions, early, fasting, and not have people, have people relax before, because acute stress mobilizes cells and changes cell distributions and likely changes telomere length. And then, unfortunately, I think Dale will talk more about this, the measures of telomere length across labs vary widely and are not comparable at the moment. There are some efforts to fix that. So in summary, telomere length appears to be a good index of exposures broadly, exposures to early life stress as well as current stressors. It's associated with prenatal conditions and current psychological disorders and lifetime exposures. So really, when we look at telomere length, we're looking a little bit at a measure that's historical as well as current. It's sensitive to duration of exposures, and this, I think, is an interesting point for exposure science. Studies have shown that duration, well-measured duration of depression, exposure to violence, exposure to years of obesity, as Dale will show, and years of caregiving, all are associated in a dose-response way with telomere length. So what are some of the advantages and limitations of using telomere length? It's really a foundational marker. It's the basic unit of how cells age. So it is associated with many, many things. It's not an indicator of stress alone. It's an integrative marker, as Maria said, of genetics and environmental exposures. So we can't make strong inferences about one particular factor. It may be useful if multiple exposures are measured and examined, and that way we can make sense of a question like, is lead exposure associated with telomere shortening? If we're measuring several of the other big factors that we know are correlated with lead exposure and are both historical and current. And then we can look at additive relationships and interactions. So really we need to move beyond bivariate relationships and cross-sectional relationships. Telomere length, it predicts multiple outcomes. It's not a specific marker of cancer or cardiovascular disease. So that could be a disadvantage if you're only measuring one disease and you don't find a relationship, but it's an advantage in that it appears to be a building block of an underlying factor in diseases of aging broadly. So it's powerful in predicting the spectrum of aging-related outcomes. And I am at my limit of time. I just want to mention I really feel like we're at the tip of the iceberg in understanding how useful telomere length is and what it means when we measure it in humans at any point in the lifespan. So this is really an unfolding story that's a rapidly growing field, and I am excited to see how it fits into the exposome and what people find once they start looking at ADEX and other markers of chemical exposures. This is our website at UCSF that lists most of the studies I showed. And here are some of the main players and collaborators that I'm very fortunate to work with. You can see at the top Elizabeth Blackburn, and under her Yuxia Zhulin, and they have been the main driving forces in being able to implement telomere length into these different clinical and population-based studies. Thank you so much, and I'm happy to take questions.
Okay, so we have a time for about two to three questions.
Hi, John Moran, Michigan. That was a very provocative talk. So I guess the question I have is with social situations, it seems like there could be a lot of confounding variables. And for example, how accurately can you measure stress in a mother from a historical self-reporting? And then when you're assessing telomere length, are those experiments done blindly? Basically, I'm looking for some of the details to, how the experiments were done that in some of the key citations that you listed.
Right. Those are both good questions. In terms of measuring stress retrospectively, I don't think any stress researchers believe we can do that measure stress perception retrospectively in any accurate way because it's so influenced by current mental state. What we do measure retrospectively with some level of accuracy is exposures, events that happened, like a death in the family, a divorce. So those early exposures that we measure from the family environment, like were your parents divorced, were they alcoholic, etc., those tend to be reliable. The only one that's not reliable is reported sexual abuse. And that one is, there's some inaccuracy in that, but it's still important to measure it. That's, I think, the conclusion. So stress, the more specific we can measure an exposure, and that means it has to be major and very a definable event. That's what we try to measure in our stress measurements, and we're working very hard on better retrospective measurement of a cumulative life stressors. And George Slavich at UCLA is developing this measure that we've been using, and it's called the strain. And it's exactly the point. It's trying to get at better accuracy of recall by not having a lot of room for subjective input. And your second question was, how accurate are these telomere length measures? How biased might they be by knowing which group is the exposed group versus the control group? So in our studies and most people's studies, the lab that's measuring telomere length is using de-identified samples where you can't tell from the ID number what group the subject is in, if they're in a treatment group or not. There was recently a valid study looking across different assays by Abraham Aviv and Liz Blackburn that did a very well blinded study sending the same samples to various labs and looking at concordance between the assays. The concordance between Southern blot and PCR was pretty good, but there are some differences, and I refer you to those papers.
A question on the assay.
Okay. George Daston, Procter & Gamble. Thanks for a really thought-provoking talk. I wish I could keep you here all day asking questions, but I'll just limit it to two. One is methodological, which is how good is the correlation between telomere shortening in the accessible tissues that clearly replicate a lot versus tissues or cells that don't replicate much or at all? And then the second is, what's the heritability of telomere shortening? I guess I was thinking through your talk about, some of these chronic disease or stress markers associated with with telomere length, and then just wondering about what the long-term effects of, say, World War II has been. So those are my two questions.
Great questions. So the first question about how telomere length and replicative tissue is associated with, tissue, non-mitotic tissue that doesn't divide. That's an excellent question. So I can refer you to, I can't answer that directly, and I don't, I know that in studies of newborn tissues, there's a high correlation across tissues, post-mitotic and mitotic, but that's newborns. I believe that correlation, it probably changes dramatically with aging. There was one study that capitalized on the point that non-dividing cells will reflect telomere length from birth more easily, and dividing cells should reflect biological age. And it's the comparison between a dividing blood cell, average telomere length, and a muscle cell telomere length that should be very informative. And this was just one study, but it was super interesting. It was by Abraham Aviv and colleagues, and they looked at dog longevity. And they found that the difference between if I recall correctly, blood telomere length and muscle cell telomere length predicted longevity in a very linear, strong fashion, and that just blood cell telomere length alone was much weaker. So that, if only we could easily get post-mitotic tissue in humans, we might have much more predictive models.
Okay, thank you for that wonderful answer. I have to apologize to everybody. I have to stop the questioning period and hold. If you can hold your question, because we're going to have a very long discussion period. So maybe, particularly if Elisa is coming back, so we're going to wait for her answer later on.